Okay, well, see how things go. This. Okay, you guys get to make some decisions on behalf of your class, so make some good decisions, I guess. So have you thought of the 3 topics that you actually want to have tested for the cumulative section? Do any of you want to share what choices you have? Yes, Jamila. Skeletal muscle, okay. Is everyone really strongly opposed, like I hate skeletal muscle, I never want to see it again? Okay, any other choices? Yes? The nervous system? You do know I'm a neurophysiologist though, right? So I could make it like really, really hard. Yes. Special senses? Just the eye? Okay, are there any other choices? Hormones? Simple hormones? Any other choices? Hormones is up there. Yeah, so just remember taste. Remember all those the 5 different ways you taste? You remember them all still? Umami work. How does sweet work? Okay. Okay. All right. So out of these 4 choices that you guys have, I will post them online somewhere, also on social media, and you guys will pick 3 out of these 4. And then once you pared those down by Friday, then I'll come up with individual slides that you need to know from those sections. Sorry? The respiratory system. The lungs? No lungs? No? No? I think you're being outvoted already on that one. So hormones, skeletal muscle, the nervous system, and then special senses, and we'll draw questions from there. Yes. But receptors? Okay, so we'll draw from a choice of these 5. You guys will come up with 3. It'll be majority choice. Okay. All right. Now that that's out of the way, so the only other thing I'm going to mention is because of religious observances around Ramadan, and I want to make sure that anyone who is observing has the opportunity to spend time with their family and to take part in iftar. We're going to try to end today's class a little bit early. Anything that I don't have covered, I'll make a recording of. And then Wednesday's lecture, because I hadn't thought of it at the time, I'm just going to do the same thing as I'm doing now. It'll be available for you on Tuesday evening, so there will be no formal lecture here, and you should just view it on your own time, okay? And I'm going to be doing it all at U of T, so hopefully there won't be any issues of things cutting in and out, because I'll just be doing it in my office, and I'll just use a hardwired Ethernet connection. So we're going to go through this section, which is on your final exam, related to fluid electrolyte balance as well as the renal system. And I want you to start thinking a little bit about this whole system, because the first part, actually, you need to and the different fluid compartments. If you haven't studied them before, it's a good time to go back and review it. It's also really important because we deal with isotonic fluids. So fluids that don't have any kind of osmotic gradient. And so one of the things that, for example, our sweat has a certain number of ions that are associated with it. So all those sports drinks that you guys take in, a lot of them actually designed to either be isotonic or to be hypertonic, of all things, to allow you to rehydrate appropriately. And there's a lot of science behind all this. And we're going to go through a couple of the different fluid compartments. We're going to be looking at some of the different mechanisms that allow you to maintain these fluid levels, including coming back in the second part to go back over the renal system and make sure that you understand how the kidneys filter the blood, and then how different hormones are involved in maintaining these electrolyte balances. And as we go through these different systems, one of the things to make note of is that your body has inherently some different buffering systems, and we're going to try to make sure that as we go along, you know the different types of buffering systems and the main ones that are parts of different compartments. Do any of you know who this individual is? She's no longer with us. She's from San Diego. Any ideas? In the context of what we're talking about with fluid electrolyte balance, especially nowadays when you guys might have taken part in heart and stroke or any of those other marathons, the walkathons, etc. Especially at this time of year, it's really important to take into account the story behind Jennifer Strange. Do any of you know Jennifer Strange? No? 2007, what was really big in 2007? Sorry? Sorry? Swine flu? Okay. Pop culture big in 2007. 7 years ago, what was like the hottest thing? What's the hottest thing now? Nothing. Sorry? Nothing. Okay. Nothing is hot now. Okay, awesome. 2007, one of the hottest things that was available, and this was back in December. This was in San Diego. This was part of KCND, which was a radio station. What was really popular back in 2007 was the Nintendo Wii. Have you guys heard of the Nintendo Wii? Yes, it was like the big thing. Everyone needed to go out and get this new Nintendo Wii. It was a big thing. It was flying off the store shelves. No one could get it in time for Christmas. And Jennifer Strange was one of those individuals who heard about this radio contest down in San Diego. And one of the contests involved whoever could drink a certain amount of fluid and then not go to the washroom, whoever could hold out the longest would win this Wii in time for Christmas. And it was called Hold Your Wii for a Wii. And Jennifer Strange went and took part in this contest. She didn't win. There was another individual who did. She drank several gallons of water, and you can see her actually taking part in this contest here. And then at the end of it, because she didn't pee for a while, she went home and she started having headaches. She had a headache, led to a coma, and eventually she died. She died of a very classic case of what's known as water intoxication. You can literally kill yourself with water, and those are one of the things, especially now in the summer, where you keep hearing about hydrating yourself. And it really is important to hydrate yourself, but you can actually overhydrate yourself. And it's one of the things that I want you to make sure that you are aware of, and we're going to come back to water intoxication in a few moments. So this is a really important slide. I don't need you to necessarily memorize all of these different numbers, but I want you to start thinking about this. Your body weight, 60% of it is due to water. Your body has a lot of water. When you're gaining weight, you're actually gaining water. When you're trying to lose weight, for example, if you're exercising, one of the things that you blow off is actually water, which is one of the ways in which you lose weight. So if you're taking a look at the total distribution of total body water, and let's say you're a 70 kilogram individual, and that means about 60% of your body weight is water. Let's say you have about 42, or in this case, 40 liters of water. How is that distributed? So if we were to take a look, we'd actually see that the extracellular fluid, the fluid that surrounds the cells, so things like the interstitial fluid, so like if you were to open up the different tissue organs, if you were to open up the gut, all of the fluid that is surrounding the different organs, surrounding the different cells, the extracellular fluid would add up to about 15 liters. And that can be further broken down into interstitial fluid, the tissue between tissues, fluid between tissues, and then the plasma, the fluid that actually runs from one part of your body to the other, supplying the different nutrients, etc. So again, this is extracellular fluid. Interstitial fluid is part of it. Plasma is part of this. And again, it's a very small component. The vast majority of the fluid in your body is intracellular fluid. What does that represent? 25 liters of intracellular fluid. What is intracellular fluid? Okay, so this is inside your cells. So if you were to take every last cell in your body and treat it like a little grape, and you were to squeeze out all the water from every single cell in your body, inside the cell, you would be able to capture about 25 liters. It's a lot. Each cell is by itself very small, but overall, because we have so many cells and they're all packed with water, it means that we have about 25 liters. The way that we look at water and body fluid components is that we usually look at how it goes from plasma to and then from interstitial fluid, and then from interstitial fluid, how it equilibrates to the intracellular fluid. You can actually blow cells up, which is what happened to Jennifer Strange. She had a headache because the neurons in her brain were starting to swell. The water that she had drank that got into her plasma, that got into the interstitial fluid, is capable of crossing the blood-brain barrier and capable of actually getting into the neurons, and the neurons were swelling. They have nowhere to go, and so she died of a coma. Again, that slide that I just showed you is really important one. And this gets into the importance of water. It's a universal solvent. We don't really need to worry too much about all of the different solutes, although we have some non-electrolytes and some electrolytes. Most of the non-electrolytes tend to be organic. So these would be in things like glucose, lipids, creatinine, and urea. And we're going to see all of these later on today. These don't normally dissociate in water. They're just held in suspension in water, versus electrolytes, which form different types of ions. And we have a lot more of these electrolytes as solutes than we do these non-electrolytes. So we have more sodium ions than we do glucose molecules. And as a result, because these are the most abundant form of these solutes, this tends to form and create this osmotic power that we talked about earlier. How do you move things from one compartment to another? It depends a lot on these different electrolytes, which gets us into going back to the extracellular fluid versus the intracellular fluid. Again, the intracellular being if you took every last cell, treated it like a little grape, and squeezed out the contents of the intracellular fluid. You already know a lot of this from the lectures that we had on the nervous system. The extracellular fluid, the ones that we have around the cells, we have the major cation. They're the major solutes. We have a lot more of sodium, and the major anion is chloride, right? Surprising? Probably not. In the intracellular fluid, though, we already know that we have very, very low levels of sodium inside the cell, and we have very low levels of chloride inside the cell. So ionically, in terms of the solutes, even though we have water inside the cell and water outside the cell, the fluid compartments have a very distinctive pattern from the inside versus the outside. Okay? Again, just to keep all of that in mind. So in terms of how water moves from one compartment to another, so we look at those three different compartments from plasma to interstitial fluid and then into intracellular fluid. This water moves across those different compartments freely through osmosis. And again, the osmosis can be both ways. It can go from one compartment like the intracellular back into the extracellular fluid compartment. And when we're talking about changes in ionic concentration, we're often looking at active transport mechanisms or how different channels allow flow across different membranes. If we're changing any of the solute concentrations, which again mainly means electrolytes, which mainly means ions, that means that there's going to be a change in the way and the direction in which this water will overall be flowing. Again, you don't have to worry about memorizing everything on this particular diagram. It's just to give you an idea that things that we've actually looked at, like the lungs, the GI tract, etc., allow for different types of molecules to go from one compartment to another. So oxygen and carbon dioxide get into the plasma, which eventually gets into the interstitial fluid around the cells, and then we want it to get inside the cells. Same thing for the GI tract. We learned about how different nutrients get formed, how they get broken down, and then once they're there, we learned how they go from different parts of the lymph and blood eventually into the interstitial fluid, which allow different nutrients to get inside the cell. You can see that there is often a one-way direction for these. So nutrients usually go in, water can go either way, but carbon dioxide usually goes out of these cells. What we're going to be talking about today is this last system, which is the kidneys, and how we can look at the water as well as the ions that you take in, mainly through the systems that are here on the left, and how we have to equilibrate all of these in the blood plasma, and in the interstitial fluid, and in the intracellular fluid. So if we don't do this, then when you're ingesting fluid very, very quickly over a small period of time, or you're not taking in any fluid, and again, some of you are fasting, so when you're not taking in fluid, adapting, because if it doesn't adapt, then you run into all sorts of problems. And that's, again, just an overview. So you don't have to worry about memorizing these numbers. Usually, for an average individual, the amount that you take in per day is equivalent to the amount that you actually generate per day. So again, through metabolism, your cells undergoing oxidative phosphorylation, one of the byproducts of that is actually water. And these water molecules that you form in each of the cells adds up to about 250 mils per day. Your food intake is about 3/4 of a liter, and then the amount that you drink in is the vast majority. You're losing things through your feces, your sweat, through your skin and lungs, as well as through your renal system and through the urine. But overall, we have no net gain nor net loss. If you're gaining weight, for example, often just cutting down on the amount of beverages and the amount of fluid that you're taking in is enough to offset any weight gain that might occur. So again, those few slides are not important. You need to go back over the slide related to the overall sizes of the different compartments and how water moves between those different compartments. But this slide and the next sort of slide is important for you to make sure that you know. I think in your notes, the arrow on the plasma osmolality is actually going the wrong way. It should actually be an increase in plasma osmolality. And again, this is mainly due to the type of food that you take in, etc. So I want you to understand what you actually drink. The thirst mechanism is the major reason why we take in 1.5 liters of water every single day. This involves osmoreceptors that are found within the hypothalamus. So I'd like you to make sure that you know that the thirst center is centered around the hypothalamus. It involves osmoreceptors. Any type of increase in plasma osmolality will increase the activation of the of the osmoreceptors. If you have a dry mouth, that's enough to stimulate the thirst centers in your hypothalamus. If you are losing too much blood, or you are losing too much blood pressure, one of the best ways to actually get this back up, so some of you might be interested in medicine or paramedicine, if you are going into shock because your heart simply doesn't have enough fluid to be able to pump around. So if you've had a gunshot, you've had a stabbing, you've lost a lot of blood for whatever reasons, you don't have enough blood to go around, this can lead to shock. And one of the things that happens is, even before they'll give you like a blood transfusion, is just to increase your blood volume in order to increase your blood pressure, and they'll put in a saline or a glucose solution via IV. So all of these different factors I'd like you to make sure that you are aware of. This is sort of also found on the next slide, where a change in plasma osmolality will decrease the amount of saliva production, leading to a dry mouth. Will also stimulate the osmoreceptors in the hypothalamus. If for whatever reasons you start losing plasma volume, you'll drop your blood pressure, and then this can also lead to the production of angiotensin. So for the time being, you don't have to worry about all of these. This is more of a synthesis slide. And ultimately, this leads the hypothalamus to start firing. It makes you want to drink more. You take in drink, and it kind of balances all of this out through negative feedback. I don't think I actually mentioned this explicitly. Where does actually water get absorbed in the GI tract? Small intestine, it'd be a pretty good bet. Large intestine, we also do absorb water from there. Do we absorb any from the stomach? Nope, nothing. The stomach is not really a big source of absorption, so it's the intestines and the large intestine. The stomach is really only good for absorbing. If you take it in, some drugs can cross from the stomach directly. And any guesses? what do you get into your bloodstream incredibly quickly? What kind of things that you take in? Into your bloodstream fast if you eat it, drink it, whatever. Glucose? No, not glucose. Caffeine? Not caffeine. Good guesses though. Even faster. Sorry? Hormones? You drink and eat hormones? Okay. If you do that, probably not. No, they don't work that way. Alcohol. So alcohol gets through the aspirin. Alcohol, there are very few substances that actually get absorbed directly from the stomach, but alcohol is one of them, which is why, for example, people get inebriated or have intoxication a lot faster because it gets absorbed directly from the stomach, doesn't have to get down into the intestines at all for absorption. So we're going to eventually be talking about this. You don't have to memorize this slide now. It's more about trying to get you to think about how we maintain the homeostasis of our water. We have a number of different hormones that come into play. So we have a behavioral consequence on the previous slide. We also have this antidiuretic hormone, or ADH. This antidiuretic hormone is what we're going to be concentrating a lot on later. You don't have to really worry about all of the decreases that you see here, nor do you have to worry too much about this at this time. It's just to give you an idea that one of the things that happens is that when a signal is sensed and we release antidiuretic hormone from the posterior pituitary, so whoever is a big fan of hormones, no, not a big fan anymore, right? Anterior pituitary, posterior pituitary, it's like one of the most common multiple choice test questions that you'll be asked. Which of the following is released from the anterior versus posterior? The posterior pituitary releases antidiuretic hormone, which then acts on the kidneys to cause water retention. So it's antidiuresis. Diuresis meaning that you are releasing, in terms of your urine, you have lower urine output when you have ADH around. So you have very small urine volume. So the opposite way, so you can also have dehydration. This is where you're in a negative fluid balance, and you can have a loss of the extracellular fluid, either due to things like hemorrhage for a variety of reasons. You could be in an accident, you could be losing lots of blood, you could have a severe burn. This is another way in which you lose a lot of extracellular fluid. If you have prolonged vomiting or prolonged diarrhea, especially if you are not capable of ingesting correctly, so young and old people, one of the reasons why diarrhea and vomiting, especially in the very, very young, infants or older individuals, this becomes a problem, is because there is a negative fluid balance. You are not taking in enough fluid, you're losing too much, either in your diarrhea or through vomiting. If you're sweating an awful lot, or you are engaging in a voluntary water deprivation, which again, many individuals are right now because of their fast, then this water deprivation all lead to this negative fluid volume. So some of the different signs and symptoms of this include things like having thirst, of course. Obviously, you get a dry mouth from this. Your skin will become a lot more dry and flushed, so it'll look like there's more blood flow going through, as well as oliguria. What's oliguria? Any ideas? Can you make a guess? Obviously, it has something to do with urine. Any guesses on what you're going to do with your urine? Yeah. If it's yellow, sure. Dark yellow, which means what? You're on the right track, but what does that mean? Yes, but when you have like, this is going to sound really gross, but we've all experienced this, right? So if you've drank a lot and then you go to the washroom, is your urine dark yellow or lighter color? It's usually a lighter color. And do you produce lots of it after you've drank a lot? Usually you do. So in this case, you're producing a very concentrated dark yellow urine. So smaller amounts of urine, right? So again, this may lead to things like weight loss, depending on how long this is going on. It could lead to fever, which is again one of those really big signs that everyone is looking for with water deprivation. If you can't control your body temperature, it becomes a huge issue. Mental confusion, oftentimes individuals who are out dehydrated, they just won't be able to answer questions. It almost looks like they're having a bit of a stroke, to be quite honest. Hypovolemic shock, and of course they might start losing their electrolytes. Another way to talk about what happened to Jennifer Strange, and I'd like you to make sure that you know this slide, as well as some of the signs and symptoms associated with hypotonic hydration. It's also called cellular overhydration. It is also called water intoxication. Down here at the bottom, it's literally where you are drinking yourself to death, and for the first little while you actually look like you're drunk. You lose a little bit of coordination, etc. It can occur for a variety of reasons. Your kidneys might be failing. That's one reason to actually get cellular overhydration or hypotonic hydration, etc. Water intoxication is a special case where you are specifically drinking lots and lots of water over very, very short periods of time. So I don't know if you can actually even do this, but if you had like about liter bottles of your favorite soft drink, would you be able to drink all of that within 20 minutes? Water, for example, that Jennifer Strange was drinking and then holding after she drank it all. So this again is a form of water intoxication, and what happens is that the ECF, the extracellular fluid, because it's in balance with the plasma, so the extracellular fluid eventually becomes more and more diluted. What is the major ion that we find within the extracellular fluid? And chloride. So one of the more important ions is sodium. And what happens is, because the amount of sodium stays the same, but the fluid that it's actually dissolved in as a solute becomes bigger and bigger, you end up getting hyponatremia. You have low sodium in the extracellular fluid. And the net result of this is that you get osmosis into tissue cells. You're trying to get that water out. The only other place that it can go is back into the cells, and this causes the swelling of the cells. And this can lead to severe metabolic disturbances depending on where it is. So if it's up in your brain, it might lead to nausea and vomiting as some of the receptors and neurons within the brainstem start to swell. If it's in your muscle, it might lead to muscle cramping. And again, it can lead to cerebral edema or a swelling of the brain as well, and possibly death. And this again happens almost every year, at least once or twice in Canada, especially during the summer where people are out exercising, enjoying the good weather, trying to stay hydrated, and they overhydrate themselves. They don't take care of themselves, and they eventually cause all of these different symptoms leading to death. If we talk about edema, there's a couple of things that I want you to be aware of that causes edema. So if you're asked the question, what are some of the causes of edema or the abnormal accumulation of the interstitial fluid causing tissue swelling, this is anything that will hinder the blood, the flow of fluid out of the blood, or to hinder its return. So if you have increased blood pressure, you often have edema, right? This often takes the place in things like congestive heart failure. For those of you who know individuals with high blood pressure leading to congestive heart failure, what is one of the big signs of edema in those individuals? Where do you get tissue swelling in individuals that have high blood pressure for long periods of time that lead to congestive heart failure? Any ideas? What part of your body do you think swells? Your kidneys? Possibly. How about on the outside? Like your head swells? Your arms swell? Yeah? Your limbs? Okay. So what's the most logical place for a limb to swell? You're on the right track. Again, think like a physiologist. Where are you going to accumulate fluid the most? So if I So what do you do when you have swelling? What does your doctor tell you to do? Your doctor doesn't tell you to do anything. You're going to the wrong doctor. So you have swelling in your leg. What do they tell you to do? Elevate it. Okay. So what's the lowest part in your body? Your feet and ankles, right? So with increased blood pressure, often you see edema in like the ankles of individuals that have high blood pressure. If you ever sat there on the subway and you took a look at someone's ankles where you can't see them anymore, they likely have congestive heart failure because of increased blood pressure. They probably also have increased capillary permeability because they also have inflammation that's systemic. You might also have this because anything that hinders blood return could be incompetent venous valves, could be a local blood vessel blockage, and I just told you about congestive hypertension is the same as blood pressure, or you could have increased blood volume. Okay, so all of these different things can cause edema, which is this atypical accumulation of interstitial fluid. So again, other things not quite as well known might be where you have a change in the colloid osmotic pressure. If you don't have enough proteins within the interstitial fluid, with the decrease in plasma proteins, this might also cause edema as well, but it's a lot less likely that this will occur because you would have to suffer from something like protein malnutrition, where you're not taking in any protein sources, no protein in your diet, meaning that you have no protein in your plasma. Again, very, very rare nowadays because even if you're a vegan or vegetarian, you have alternate sources of proteins. You could have liver disease because the liver is one of the sites for protein production, or you could have inflammation of the glomerulus, which is part of your kidneys. So some of the things that might cause this might be if you go in for a surgical procedure and they're cutting, or they're making an incision in your skin or somewhere in one of your tissues. We didn't really talk about the lymphatic system outside of the digestive system, but the lymph system actually helps to return a lot of the fluid. If you cut some of those vessels, then you can actually cause the leaked proteins that come out of the lymphatic system to accumulate within the tissue. And again, in some surgical procedures, that can definitely happen. Okay, so if we go into the electrolyte balance, again, I mentioned this in passing. Electrolytes that we looked at were mainly salts. We're going to also consider acids as well as bases. Usually we refer to salt balance because it's a lot easier to talk about electrolytes being salts. And when you hear about Gatorade or Powerade, we're talking about those electrolytes being things like sodium, magnesium, zinc, etc. And again, we look at salts that enter the body via ingestion, but tend to be lost through a number of different routes like perspiration, feces, as well as through urine. So we know that they're really important in controlling fluid movements from one compartment to another. So one compartment, water tries to balance itself out by osmosis across the different compartments. We've already looked at that. We can look at muscle excitability. If we don't have enough calcium, we can't get contraction. Not enough sodium, we can't get the nerves to react, etc. They're important in secretion, and they're also important for membrane permeability. So we need to make sure that our electrolytes are always in balance. I've already mentioned this before. Sodium is the most abundant cation within the extracellular fluid, and within the extracellular fluid compartment, sodium contributes to most of the solute concentration. If you get nothing else from this, the next few slides, you should make sure that you know what the major types of acids are, the buffering in the different compartments, and the major ions. And this would be an example of a really easy question. So I'm hoping that if we have that on there for your final, all of you will get that perfect. Okay, so One of the things I mentioned to you is we're looking at how we regulate some of these different things, not just the fluids, but also the different electrolytes. And if we're looking at sodium, we can actually regulate this in a couple of ways. And one way that we can regulate this is with aldosterone. Another one is through ANP, or atrial natriuretic peptide. When we want to reabsorb sodium, most of this, 65%, about 2/3, is reabsorbed at the level of the kidneys in the proximal tubules. About 25% of this can get reclaimed in the loops of Henle, which means about 90%. And if we want to recover any more, we can use aldosterone to do the active reabsorption of the remaining 10%. So we are pretty good reabsorbing sodium because it's so vitally important to us. But if we really, really need to get more sodium back and reabsorb it, because it's all getting filtered at the kidneys, everything gets lost at the kidneys, and then we use different parts of the kidneys to reclaim or reabsorb some of these. And again, we'll go into ADH in a moment. Do you guys have any questions about this before we go on? Just out of curiosity, have you guys ever talked about the kidneys before, the structure of the kidneys, the nephrons of the kidneys, a little bit? Where did you learn about the nephrons of the kidney? High school. Okay, so you've learned some of this previously. Okay, so in terms of aldosterone, an increase in potassium, or if we are finding that we have low levels of sodium in the blood plasma, this will stimulate the adrenal cortex. The adrenal cortex in turn releases aldosterone, which then targets the kidney tubules to cause increased reabsorption of sodium and to secrete potassium at the level of the collecting ducts as well as tubules, eventually causing a negative feedback inhibition of this whole pathway. So again, thinking about this a little bit, maybe it's helping to make up your mind a little bit. Hormones, good. You're good on this. Remember we have like the cortex and the medulla on the adrenal glands. You guys remember all the different hormones that are released from the cortex versus medulla? So maybe rethink that a little bit. I think you guys are all good with nervous system, maybe skeletal, maybe special senses. Pick your three very carefully so you know, you get the best possible result on your final exam. The other molecule that's involved in regulating sodium balance is atrial natriuretic peptide. This is released by the atrial cells, and this is a slide I'd like you to actually make sure that you circle and come back to revisit later on. So the most important thing from this slide is really the effect on sodium reabsorption. I'm hoping that you'll keep that in mind. It's really where the hormone actually gets released from. So it's the adrenal cortex that releases aldosterone. And in this particular case, what we're looking at is this hormone that we call atrial natriuretic peptide is actually released by cells within the atrium. So within the heart is increased blood pressure, or when there is increased blood volume, one of the things that happens is the atrial cells stretch more in response to the increased pressure, and they release atrial natriuretic peptide. And one of the things that it does is it helps to decrease the blood pressure as well as decrease the blood volume. And the main mechanism that I'd like you to make sure that you know is it does so by increasing the secretion of sodium. If you get rid of sodium, water always follows. Wherever sodium is going, usually water follows after it. Okay, so one of the reasons why people with a high sodium diet tend to gain more weight is really because as you sodium, then you are also bringing water along with it, and water is one of the ways in which you increase your body weight. Okay, so again, we won't worry too much about memorizing all of the details here. If you're really interested in it, please go through it and you know, try to get as much out of it as you want to. But for me, the most important things are where this gets released, what sort of stimulus causes the release of atrial natriuretic peptide. And again, the most important thing is that we don't try to reabsorb this. This actually causes sodium to be released from the kidneys, and we lose this in the urine. And because water is following after it, this decreases the blood volume, which means that we decrease the blood pressure. Okay. So another really important electrolyte is this potassium, and this potassium, the importance of this potassium has been the way in which we've looked at excitable cells. Do you guys remember what RMP stands for? Resting membrane potential, especially for neurons as well as muscle cells. Cardiac muscle cells in particular make use of this as well. If we're increasing the extracellular fluid concentration of potassium, you actually reduce the overall resting membrane potential. You make it more likely that the cell is going to depolarize. And you can work this out if you want through looking at the driving force for potassium. Usually it is a lot higher inside than it is outside. Now there's a lot more outside than there is inside. If you increase it, you're actually going to cause potassium to come inside the cell, and this will change the resting membrane potential, and it will cause depolarization. You can also decrease the extracellular potassium concentration, which will hyperpolarize the cell and make it less likely to respond. Again, we can look at some different hormones that are involved in this, the most important one being aldosterone, that I'll ask you to make sure that you know. If we have aldosterone that we looked at with sodium, this also stimulates potassium secretion later on as well. Okay, so we'll come back and revisit this when we get to the kidneys, but for the time being, I'd like you to make sure that you know aldosterone also causes the loss of potassium in the renal system as well. One other really important extracellular electrolyte is this calcium. Again, really important for excitability. We didn't talk about blood clotting, but it's really important as an initiating factor in blood clotting. The cell membrane permeability, the secretory activities, all of these things require calcium. So we're going to look at some of the things that regulate calcium. So the biggest issue is, and this is the one that I'd like you to be more familiar with, if you have hypocalcemia, you can actually change muscle excitability as muscle tetany. Hypercalcemia can actually alter neuron and muscle cell function as well, may also cause heart arrhythmias. We've already learned this before, so maybe, again, not to beat this over the head, but these are other hormones. You guys all remember these hormones? Now, but you do remember learning about these hormones. You just can't remember what they were for. Okay, so maybe again rethink the whole let's do hormones on the final exam. The calcium balance gets controlled by two different hormones, calcitonin as well as parathyroid hormone. And we actually went through parathyroid hormone earlier in the endocrine and hormonal section. And again, you can go through this if you wish. I'm not going to test you other than knowing you want me to, other than knowing that these are the two different hormones that actually are involved in calcium balance. It's not like you're going to have less than 450 slides to go through anyways, right? You can do that. It's like it's an easy afternoon of memorization. Yeah. Okay, so a couple of other things, then we'll take a really quick break. What I'm going to probably do today is I'm trying to probably go through about 60-ish slides. The remaining 30 slides I'm going to probably put online for you. So we'll probably wrap things up, if you guys don't mind, at around 7:45 to 8 o'clock, and then the remaining slides I'll just post for you online as a recording, just as I'm doing right now. So a couple of really important considerations for definitions. So when we look at acid-base balance, when we talk about how different electrolytes and how different types of ions are dissolved as solutes within the water compartment in the different tissues, whether it's ECF, interstitial fluid, or whether it's in the intracellular fluid, the pH has a number of different effects. One of the reasons why you learn about pH in your biochemistry classes is because it has dramatic effects on how different proteins function, whether or not they're functioning effectively, and will affect all kinds of biochemical enzymatic reactions. So when you have alkalosis or alkalemia, these are somewhat interchangeable. Alkalemia refers to levels in the blood. Alkalosis is a general term about having an alkaline uh, disturbance. This is where we measure your blood level of your blood pH, and it's over 7.45. So anything over 7.45, you have alkalosis or alkalemia. Anything under 7.35, you have acidosis or acidemia when your arterial pH is below this. And this means that we normally don't go outside those ranges. Okay, so it would be very, very unusual unless you're sick and you're in the hospital that you would be outside of those ranges of 7.35 to 7.45. You have a very narrow range of pHs because your body is adapted to a very narrow pH range. So we have a number of mechanisms that actually help to maintain this pH range the best that we possibly can. So the concentration of the hydrogen ions, which is related to pH, is regulated sequentially by first the chemical buffer system. And this is a slide that I want you to make sure that you know. The chemical buffer system is rapid. It's the very first line of defense. The next system that is there in place to look at any changes in pH are the brainstem respiratory centers. If you have a change in your pH, blood pH, whether it's too low, too high, your brainstem activates respiratory centers that detect this pH change and will react within 1 to 3 minutes of any pH change. And then the last mechanism, and one of the reasons why we're studying the kidneys, is the renal mechanism. Is by far the most powerful, but it actually requires several hours to days to readjust your blood pH to get it back in the normal range. So we have one that tries to neutralize things at the very top as quickly as possible. If it's not effective and it doesn't work, then the brainstem respiratory systems kick in. And then if those aren't working well and we still have elevated pH levels or decreased pH levels, then we start looking at what happens in the kidney. So these chemical buffer systems that take place almost immediately, the ones that are the first line of defense, are things like the bicarbonate buffer system, the phosphate buffer system, and then the protein buffer system. So the next 3 slides are ones that I want you to spend some time over, making sure that you know the major characteristic features of all of these. So again, it's actually highlighted here for you. So of those 3 chemical buffers that react almost immediately to any kind of pH change, the bicarbonate buffer system is the one that helps to buffer both the intracellular and extracellular fluid, but within the extracellular fluid, this is the only important extracellular fluid buffer that is a chemical. Okay, so we need bicarbonate in the extracellular fluid. What other 2 compartments make up the extracellular fluid? Because if you know this, then the multiple choice test question would be really easy. So is bicarbonate system or an important buffer system for the blood plasma? Yes, no? Yes, because plasma is part of the extracellular fluid compartment. What is the other fluid compartment that makes up ECF? Interstitial fluid. So if I were to ask, is bicarbonate a really important part or a buffer within the interstitial fluid? Yes, it is, because it's also part of the extracellular fluid. So plasma, interstitial fluid together make up the extracellular fluid, and this buffer system is important in both. So the phosphate buffer system, the second one that we looked at, the phosphate buffer system, nearly identical to the bicarbonate. You don't, by the way, unless you're really, really into chemistry, you don't have to worry about trying to balance this equation or anything else. I think you all know what bicarbonate is, and you don't have to worry about all of these different phosphate, dihydrogen, monohydrogen phosphates, etc. You don't have to worry about those. These are the phosphate buffer systems. It'll just be given to you as a phosphate buffer system, and this can be either a weak acid or a weak base. And so as a result, this becomes a really effective buffer for any pH changes, either in the urine or in the intracellular fluid, where we actually have higher concentrations of phosphate. So we see lots of phosphate, for example, inside the cells. We see lots of phosphate in the urine, which is why this buffer system is important within, as a chemical buffer within these compartments, okay? So there's a difference, in other words. The major system in the ECF is bicarb. In the ICF, inside the cells, it's phosphate. Okay, make sure that you're completely okay with that. We'll do this one slide and then we'll take a brief break and then I'll try to wrap things up. So intracellular proteins are really important systems, and these systems are important because protein molecules are amphoteric. And you guys have all heard that term before, right? Amphoteric means that there is both a positive charge on and then a negative charge. All amino acids, all proteins are made of amino acids, and these amino acids contain a carboxyl group, release this hydrogen ion when it needs to, or it has an amino group, this Na2 group, which bind hydrogen ions when the pH starts becoming more and more acidic. And so these protein buffer systems are really very powerful buffers, and we can see them both inside the cell as well as outside the cell as well. And I think this is probably a good time to take a break, and then we'll come back and try to finish off this section. If you guys don't mind, how about I make your life a little bit easier and just take this for you? Now you guys only have a choice of Don't worry about it. Okay. Okay, you have 20 questions digested today's lecture, and then the reproductive, which will have on Wednesday, that will be online for you. And then about 20 questions, your choice of any of those three.
Resolutions for the building.
Yeah, you're going to come up with,
I did the last week.
I think we're going to probably take a really quick vote a little bit later on.
So I just have a question for the written assignments.
Yep.
I'm wondering, are you marking them or like a TA? Okay, good. Because I just submitted it today and I was checking the similarity and I wanted to make sure you know. No, because I talked to you and I told you that I already like used this topic but only had like 400 words and it was completely different assignment. So basically the similarity is on my name, my student ID, and like the references.
Yeah, that's never
Yeah,
a problem.
okay.
Never a problem. do you think, Luca? Should we go for it?
I'm against muscles.
You're against muscles? Why is stealth a good one? I have to convince you.
I think it should. I think what's worse is
Do you guys have a preference? Skeletal, nervous, and special senses.
Yeah. Sounds nice. And like we remember stuff too. For that person that training called that butter.
Yeah.
Why would you feel like a bladder explode?
Bladder is actually fairly elastic, so it's the same as your stomach. Your stomach can actually expand quite a bit. So one of the things that professional eaters do is they actually expand the size of their stomach by drinking lots and lots of water. But they don't do so like in a very short period of time. They'll just drink lots of water too. And it's the same with the urinary bladder. But yeah, she literally did feel like she had to go pee, right? Because it was pressing down on her bladder. She just couldn't do it because that was part of the contest.
So your bladder can explode?
Can your bladder explode? I guess it can rupture. Like anything can herniate. Herniation meaning that it, like a part of your internal organs can actually push through the body wall, and that can certainly herniate. And there have been lots of cases of herniation. But I don't know about, like, when you say explode, like, do you mean like
No, like not like explode, just like rupture.
rupture? Yeah, probably.
Would that be like you took that right away, or did you?
No. The bladder is a tissue that you can actually repair fairly easily. So not fairly easily, but easily. Right. I think, are we almost all back? Except for Anu. Maybe you can just get going, if that's okay, and we'll try to finish up as best we can. Oops, wrong way. Oh, also wrong. Okay, awesome. By the way, have any of you looked at your similarity scores yet? No, just Luca. That's it. So should never look at the overall originality or similarity score just by the actual score, because it means absolutely nothing. So the references could look very similar to like someone else's references, even from your own paper that you're looking at. If you submitted any other article before, like your name will come back as a positive hit. So don't worry about that. It's just if there are blocks of text that you see being highlighted. That's something that you should be worried about. You should go back and make sure that those aren't staying highlighted. Okay, so if we look at beyond the chemical buffer systems and we look at the physiological buffer systems, the respiratory and renal systems are the ones that were highlighted, the respiratory coming into play next, and then the renal one being the one that comes in at the very end. And these are slower than the chemical buffer systems, but they also have more capacity than the chemical buffer systems. And the respiratory renal system is really good at trying to eliminate volatile carbonic acid, because if you were to do the sort of equation H2O plus CO2, if we breathe off CO2 through the respiratory system, you won't be able to form the carbonic acid as easily. The kidneys are also really good at eliminating a number of different metabolic acids, phosphoric acid, uric acid, as well as lactic acid. Any of the breakdown products from lipid metabolism like ketone bodies and ketones are also going to be eliminated by the kidneys. And so they're really good at helping to prevent metabolic forms of acidosis. So again, the renal and respiratory systems are really important there. The next few slides are ones that I don't really want you to be focusing too much on in terms of numbers and everything else. I'll tell you what I'd like you to make sure that you know related to acidosis and alkalosis, either respiratory or metabolic. So the first form, respiratory acidosis, this is the normal range that we have of PCO2 levels, because again, I know how much you remember from the PCO2 and the PO2 curve and everything else. So normally the PCO2, anything that we have left over that's above 45 millimeters of mercury, because usually it's around 40-ish, can lead to respiratory acidosis. I'd like you to make sure that you know that respiratory acidosis is the most common cause of these acid-base imbalances. And it can be due to a decrease in ventilation or gas exchange, where you have a problem with your lungs, for example, you're not breathing as much as you should be, then you might have this respiratory acidosis, and it's again characterized by falling blood pH levels. The numbers, again, not being so important. Respiratory alkalosis, this is again most common where we have hyperventilation, and this can be hyperventilation where you're either in a lot of pain and you're hyperventilating, you could be under psychological stress and hyperventilating, but anywhere where your CO2 levels, because you're breathing so quickly, so heavily, and you're hyperventilating, you're blowing off a lot of CO2, your blood CO2 levels are going to fall as a result of that breathing below 35 millimeters of mercury and cause alkalosis. You breathed off a lot of acid, in other words. Remember, CO2 can combine with H2O in the different compartments to form carbonic acid. Because we don't have this, you don't form carbonic acid, and as a result, you become more and more alkaline. And the primary cause is because of hyperventilation and the respiratory system. Things that cause metabolic acidosis: if you have ingested too much alcohol, and if you drink a lot of alcohol, it can get converted into acetic acid. Another cause of metabolic acidosis could be a loss of bicarbonate, so it could be persistent diarrhea, for example. We don't this quickly enough, and this results in a loss of this bicarbonate. Could be due to an accumulation of lactic acid, it could be due to shock, could be due to ketosis and diabetes. If you're starving yourself, you can actually lead or cause metabolic acidosis and or kidney failure. All of these things related to metabolic acidosis. And the last point that I'll just cover is metabolic alkalosis. And this is where your blood pH starts to increase or rise. There are very few cases where your blood pH is going to rise because of a metabolic reason. And again, the best sort of scenario, or the most common scenario where you're getting metabolic forms of alkalosis is if you are vomiting, so your stomach is acidic, right? And the acid that's produced, if you are losing that acid that's being produced and you vomit frequently, then you might end up with metabolic alkalosis. You can also have this, you have an intake of excess bases. If you are eating lots and lots of antacids, too many antacids will increase your blood alkaline levels as well, or the levels of bicarbonate. So again, very, very interesting scenarios related to changes in blood pH that can result from a number of different causes, whether it's respiratory, breathing induced, or if it's because of a change in your metabolic state. So again, we won't worry about this slide. One of the reasons why we actually care about these pH levels, if your blood pH falls below 7, it can actually depress the CNS can result in coma and eventually in death. If your blood rises above 7.8, you can have overexcitation of the nervous system, induce extreme muscle tetany. You might start off with extreme nervousness. Eventually, it can cause convulsions. Because all of your muscles are undergoing tetany, you can actually cause respiratory arrest of your the intercostal muscles that cause you to breathe. So we do care about pH. It's physiologically really important. Too low, too high can actually cause death in different ways. Okay, so now I'm going to switch gears just a little bit, go back and talk to you about the urinary system. So we learned about why the renal system is really important. Now we're going to talk about how the renal system actually works. How do your kidneys do all these things like balance pH, regulate sodium levels, regulate the amount of fluid, and how does it recover most of the fluid that actually gets filtered through the kidneys? So I'm going to give you a really quick overview of the kidneys as the major organ for blood filtration, the basic working unit of the kidney, which are the nephrons and how they actually filter, the different regions of the nephron, which have different functions associated with them. Some are just there to filter, so that would be like the glomerulus, then the proximal convoluted tubules for absorption, the loops of Henle, as well as the distal convoluted tubules. All of them sort of have different functions. There's a specialization of the nephron, which is known as the glomerular apparatus, which we're going to have to talk about. And this is really important in terms of hormonal regulation that we'll be talking about, and blood pressure regulation. And again, as part of a recording that I'll put together for you, I'll put together a little bit about diuretics and how they work. And yes, you will be required to know these diuretics for your final exam. Okay, so the kidneys are one of the major excretory organs. So your GI tract is another one. Your lungs are another excretory organ. Can you think of a third organ that allows you to excrete things? Any other organs? Yes? Your skin is another excretory organ as well. So the urinary bladder, just a temporary storage reservoir for urine, and we had this really wonderful discussion about exploding urinary bladders and smooth muscle and the nature of smooth muscle, and how these ureters transport urine that's formed from the kidneys to this bladder, and eventually, if the bladder opens, how the urethra transports urine out of the body as urine. So these are the two organs that we're going to be spending time on. The kidneys, can you functionally survive with just one? Yes, of course, because you've often heard of kidney transplants. Do you survive well with just one kidney? No, you don't. So having one kidney is actually not great for you physiologically for a variety of reasons, hopefully some of which you'll be able to intuit as we go through things. But these ureters are where all the urine gets, or all the filtrate that forms urine gets passed along. We've stored this within the urinary bladder, smooth muscle, which means that it can expand. Eventually it can contract as a smooth muscle, and then we expel all of this out through the urethra, which isn't something that you can see well in this particular figure. So the kidneys do a number of different things, including remove different toxins, metabolic wastes, to control different ions within the blood, also regulate blood volume, as we'll see with the juxtaglomerular apparatus, regulate the chemical composition of the blood, as well as regulate pH, all things that we sort of just discussed in that previous lecture that the kidneys are involved with. So this is a diagram sort of anatomy in both in cartoon form on the right and the actual kidney here on the left. And we're not going to go through all of these different structures. The ones that I'm going to point out for you that become the most important in our discussion is this renal cortex. Again, cortex just meaning bark. So the outer bark of the kidney versus the renal medulla that you see here. So we've seen cortex and medulla as part of the nervous system. It's the same idea that we see here. There are functional differences between the cortex as well as the medulla, and I'm going to point those out for you. We form urine at the level of these different calyces. They all sort of combine and they will flow out. Once you start forming that filtrate, they will flow out through this ureter, as I mentioned to you before. Because do all of these different things, and because it filters so much blood, as you'd expect, there is a huge blood supply coming in to the kidneys. And you can see that the blood supply to the kidneys takes the place of being at this interface between the cortex as well as in the medulla. You'll see that there are no blood vessels, no major blood vessels that you can see here within the medulla regions that I'm highlighting for you with this pointer. So we have this really, really vascular, lots and lots of blood vessels coming in under really, really high pressure into the kidneys. And from the kidneys, as they come in, they will then perfuse all of the different regions of this particular kidney. Now again, I don't need you to memorize everything on this diagram. It's just to give you an overview of the tremendous amount of blood that's coming in to the kidney, that there is a very organized sort of circulatory system around the kidney, and it's coming in this particular fashion. And I'd like you to make sure that you are comfortable with how blood comes into the kidney. So it comes in through the aorta, it then comes in through this structure, the renal artery that you see here. And you can go back and look at the diagram yourself if you wish to try to follow it along. Then it gets into the segmental artery. It becomes a smaller interlobar artery. Eventually it will become an arcuate artery, and then it will go into this cortical radiate artery. And eventually, for each individual part of the kidney, this is the smallest functional unit, which is known as the glomerulus, and it goes from the afferent arteriole from the cortex into the glomerulus, and eventually the capillaries that form the glomerulus will recombine to form an arteriole. And is one of those really neat things is that you actually start with an arteriole in the kidney system and you end with an arteriole in the kidney system. We also have another set of capillaries, which are known as the peritubular capillaries, as well as the vasorecta. This will make some more sense to you in a moment. All of this is part of the filtration unit that we have. And again, in this hatched box, these are the ones that I'd like you to make sure that you're the most familiar with. Eventually, they go back and form the radiate vein, arcuate vein, interlobar vein, renal vein, and eventually we'll go back to the heart through the inferior vena cava. So the structural unit, the one that I just highlighted for you in that hatched box, are known as nephrons. And these are both the structural and functional units that actually form the urine. So if I were to take apart the kidney into its most basic units, I would find that there are about 1 million of these nephrons for every kidney. So you have about 2 million nephrons in total. And they, again, consist of two different parts. One is known as the glomerulus, which is that tuft of capillaries that form from the afferent to the glomerulus and eventually the efferent arterioles. And then the tubule itself, which is this cup-shaped capsule known as Bowman's capsule that surrounds that glomerular tuft of capillaries. So this is that glomerular tuft of capillaries that you see here. This represents this cup-shaped structure, which is known as Bowman's capsule or the glomerular capsule. And this is the basic functional unit that makes up this entire kidney. So we'd have about 1 million of these throughout the entire kidney. This structure repeated over and over again throughout the entire kidney. So again, just some nomenclature as we go through things. Because of the way it kind of bends back on itself, the part of the tubule that is the closest to the Bowman's capsule that you see here, or closest to the glomerular capsule, is known as the proximal convoluted tubule. And then it forms a loop of Henle, and then it will come back up and will form this distal convoluted tubule before forming the collecting duct. There might be another nephron sort of sitting off here that will form exactly the structure in a different way, in a orientation. There might be another one over here. All these filtering blood and eventually form urine. Another one here. So you can see these collecting ducts will eventually collect from all of these 1 million different nephrons. Any questions about this before we go on? Most important thing that you should make sure that you're aware of. Okay, so the nephrons consist of this renal corpuscle, the glomerulus, the glomerular capsule, the capillary network, as I mentioned. And one of the things that makes this unique is that it is fenestrated. It has holes in it, in other words, in the glomerular endothelium. And this is how we get things from the blood to be filtered as it goes through the capillary network and into the actual nephron that we are going to look at in a few seconds. So this glomerular capsule that we're looking at has a parietal layer which consists of these simple squamous epithelial cells. There's a visceral layer that involves epithelial podocytes. I'll show you this on the next slide, and this should say glomerular capsule, by the way, not renal tubule. And these extensions allow the foot processes to cling to the basement membrane, and then there are always these filtration slits that are part of the capsule as well. So again, here's the capsule, here's that basement membrane, here's that fenestration, those slits that we see here, and these are the podocytes that were just described to you, and this describes part of the glomerular capsule. So hopefully you'll be able to identify a podocyte as part of a glomerulus. Hopefully you'll be able to go back and review the major features of the fenestrations in the glomerulus. It's also shown to you again here. This filtration membrane is porous. It has fenestrated endothelial cells in the glomerular capillaries. It also has the visceral membrane that forms with the podocytes and their foot processes. And then there's this gel-like basement membrane. This forms the filtration membrane of the glomerulus. And I'd like you to make sure that when you're going back and reviewing later on, that this is a slide that you go back and make sure that you actually review. So why is this filtration membrane so important? So think a little bit about this, right? So your kidneys with these different units that has this filtration membrane, blood is flowing through the capillary, and blood is on one side, and we need to filter all the fluid parts of it. So all the cells, all the red blood cells, all the white blood cells, they stay within the capillary network, but all of the other things get passed through this filtration membrane. So the water, the solutes, anything that is smaller than a plasma protein actually gets filtered from the blood past this membrane into the nephrons, okay? And you can imagine if this didn't work, that you would drain yourself of fluid pretty quickly. And this filtration membrane allows for some things to be filtered, like the water and solutes, but it prevents things like red blood cells from going across, because we don't want those. We don't want plasma proteins to be going across. We want them to stay inside the capillary network. Larger molecules, bigger than things like glucose, ketone bodies, etc., also should be staying inside the blood vessel. So this filtration membrane is designed specifically to allow the fluid and the solutes, smaller solutes, to cross, but the major components of the blood, including the cells, some of the larger plasma proteins, and other macromolecules, they have to stay on the other side. So this is what is happening. This capillary, the blood is flowing by here. All the fluid component parts of this are getting through the different filtration slits and everything else, but the cells, the larger molecules, the plasma proteins are staying on this side. All the fluid components are going out this way. Okay, so your blood is being filtered, but not the cells, just the fluid parts of it. Questions on this before we get going? Yes, no, maybe? Okay, so a couple of things related to these nephrons, then we're going to come back and talk about these nephrons in some more detail. We have two different types of nephrons. We have cortical nephrons, and we have juxtamedullary types of nephrons. The cortical nephrons comprise the majority of the nephrons that we see in a kidney. So if we have 1 million nephrons per kidney, how many cortical type nephrons do we have? 150,000 of these cortical nephrons and about 150,000 of these juxtamedullary types. These juxtamedullary types are different. The cortical nephrons, everything in the nephron stays up in the cortex, everything. The proximal convoluted tubule, the small loop of Henle, everything stays up there. The juxtamedullary type of nephron have very, very long loops of Henle that actually extend down into the medulla, and they have thin segments as well as thick segments in the different loops of Henle. When we need to form concentrated urine, use these juxtamedullary types of nephrons. All of these nephrons filter blood, but for us to form concentrated urine, we actually need the juxtamedullary type. The cortical nephrons just filter blood. They don't really help us to regulate the type of urine that we actually produce. So make sure that this slide is something that you are comfortable with. Talk to you about the way in which blood flows through the glomerulus, and it goes from that afferent arteriole to the glomerulus. That forms those capillary networks, then to the efferent arteriole, and it's specialized for different forms of filtration. The blood pressure tends to be high because the afferent arterioles can adjust their diameter, and the efferent arterioles can also adjust diameter. And again, we won't worry about the differences in the size of these for the time being. We'll come back to that a little bit later on. So this is another important slide to make sure that you know. And these peritubular capillaries, which are, again, you need to keep this in context. Peritubular capillaries are low pressure. They're porous capillaries adapted for absorbing different fluid as well as solutes. These arise from the efferent arterioles that I showed you in that hatched diagram, that box on one of the earlier slides. And these cling to the renal tubules that are found in the cortex. So we find peritubular capillaries associated with those cortical nephrons. Okay? Have I lost some of you here at this point? Yes, I have lost you? Okay. Maybe I can re-find you a little bit. And I think we're going to wrap things up in a few more minutes anyways. So this is the capsule that we talked about, the Bowman's capsule. So we have the afferent arteriole, sorry, I'll do it this way. We have the afferent arteriole going in. It forms the capillary network. It forces blood through, it forces fluid through this capsule, and then we have these proximal convoluted tubules. That part is okay? And we have specialized structures that allow for filtration of only fluid but nothing else. All the plasma proteins, blood, everything, blood cell stays within the capillaries and it kind of leaves again. That part's good, right? So this is the efferent arteriole. So one of the things that happens is in the cortex, do you guys remember, do we have long loops of Henle or short loops of Henle? So they stay in the cortex. So let's imagine this is the cortex that we see up here. And the efferent arterioles will then form blood vessels that kind of wrap around these cortical, the cortical loops of Henle, which is what this is saying here. They arise, they form from these efferent arterioles, they wrap around the small loops of Henle that stay within the cortex. Is that a little bit more clear? Okay, let me show that to you in another diagram in a few moments. But peritubular capillaries that follow the tubule are actually only found in the cortical nephrons. Okay, so do we see more peritubular capillaries, or do we see less of them in the overall kidney? Which is the dominant form of nephron? It's the cortical, right? And do we associate peritubular with cortical? Yes, so we see this is the dominant form of capillary that we see in the cortex. Now again, I'll try to make this all really clear for you in the next diagram, but before we get there, We need to talk about another type of capillary bed, and these are the vasa recta. And these are very long blood vessels that run parallel to the long loops of Henle, and these are coming from the efferent arterioles that are associated with the juxtamedullary types of nephrons. Okay, and their function again is to form or help to form the concentrated urine. So these 3 points here are really important ones, and I try to go back and show you this on this particular diagram. So this diagram is actually showing you two different things. We have an area of the kidney called the cortex and an area called the medulla. So if we're looking up at the cortex, so this is the cortex up here at the top, anything under this would be the medulla. Is that part okay for everyone? So if we took a look at this type of cortical nephron, we would see that here's Bowman's capsule, and then we could see that we have very, very short loops of Henle. If we were to take a look at these structures in pink or purple, they would be associated only with these cortical nephrons. Okay, they're only found with these cortical nephrons. What do we call those? So those are the peritubular capillaries, right? And what they do is they will help with the whole reabsorption process. The other type that we see here is this juxtamedullary type of nephron. So is this the dominant type or the one that we find in the minority? So this is only 15% of all the nephrons that we see in the kidney. This is the dominant type. And so what we see here is these long loops of Henle, and you'll understand why they have these long loops of Henle in a moment. And associated with these are these specialized types of capillaries. So these are capillaries, and so are these. But these capillaries actually line up along the loops of Henle that you see here. And one of the functions that they do is to help form concentrated urine. So do any of you take Latin here at Ryerson? You take Latin here? You took it in Europe, so what's vasa recta? Okay. Can anyone help your classmate out? Can you make an educated guess? So what do you think vasa means? What does it sound like? Vessels. Yes, good association. Thank you. Vessels. So what do you think recta mean? What do they look like they're doing? Recta? Straight? Straight vessels? Because they look like they're running straight? Yes, I know you are all secretly thinking that. These straight vessels are the ones that actually form with these juxtaglomerular type of nephrons that we saw earlier. So again, just to put the things in perspective, both of these are really important for actually filtering blood because all of these are receiving blood flow. Some of these are really important because we have so many of them for forming this filtrate and reabsorbing things. But if we need to form concentrated urine, we need to have this other type, which is known as the juxtamedullary type of nephron that we see here on the right. So just to put things in perspective, the kidneys filter the entire body plasma 60 times each day, right? So every hour, multiple times. The filtrate is everything in the blood minus the proteins and the urine. So again, think of this. So you are filtering out the entire contents of your blood plasma, everything in your blood plasma, which is in equilibrium with the interstitial fluid, which is in equilibrium with the extracellular fluid volume. You're doing that 60 times every single day, and you are actually only forming 1% of the total filtrate, which means what? You are absorbing, reabsorbing all of that. So even though you're filtering so much volume, you are actually reabsorbing most of it, and only less than 1% of everything that you filter actually gets lost in the form of urine. Again, some of it very essential that we actually excrete off because it contains metabolic wastes as well as unneeded and unnecessary substances. So some of the mechanisms of urine formation include things like actually filtering things at the level of the glomerulus, reabsorbing things at the level of the tubule, which returns the glucose, amino acids, and 99% of the water, salt, and everything else from the blood. So if you're wondering, after it's gone through the glomerulus, why doesn't like the blood vessel collapse right away? Because it's almost immediately through the peritubular capillaries reabsorbed. We get 99% of the water, salt, and everything else actually being reabsorbed fairly quickly after it gets filtered through. And then if we need to get rid of things, we have this tubular secretion mechanism that we'll be talking about as well. So this is sort of an overview of how the blood pressure works to actually force the fluid portion through to keep the plasma proteins and cells within glomerular capillaries, how we then can reabsorb things at the level of the peritubular capillaries that surround the nephron a little bit later on. Again, I won't ask you to worry about all of the different values that are associated with this. We have a net filtration pressure, the pressure that actually forms the filtrate at about 10 millimeters of mercury. That number, if you're really interested, is a mathematical calculation of the blood pressure that is coming in through the glomerular capsule, and then subtracting out the amount of hydrostatic pressure going back in, etc. And the net pressure that we get is this value of 10. Have to worry about trying to calculate or know any of the values that are associated with it. If you're really curious or you're really interested, then you can come and talk to me about how these different values work. In terms of the glomerular filtration rate, if you went back and did the calculation of 60 times your blood plasma being filtered every single day, you'd actually be able to go back and calculate what's known as the glomerular filtration rate, or GFR, which says that you form about 120 to 125 mils of filtrate per every minute that goes by, and this is controlled by that net filtration pressure, the overall membrane permeability, and then the surface area that's available for filtration as well. Okay, so I think I'm going to stop here for the time being. Before I let you go, let me just ask you this, because I'm always curious. Which of the following can you not have as a color for urine? Blue? Everyone agrees on blue. Can you have black urine? Probably. Okay, can you have green urine? Yeah? Okay. Red urine? Okay. So it turns out there's only one answer on there that you cannot have. Yeah, so you can actually have red, orange, blue, green, black, and brown urine. You can have that by the types of foodstuffs that you ingest. The medication that you take can affect that. And yes, blue urine actually exists. If you take certain types of drugs like methotrexate and others, your urine will actually turn, it's not like sky blue or anything, but it turns more of a bluish tinge. The only one that you can't have is fluorescent. Your urine by itself should not be glowing in the dark. That becomes very problematic. But depending again on your diet, your health, your age, the different drugs that you're taking, you can have any of the other colors here. Some are actually normal to have, except nowadays everyone's got a lot of worries of having red tinges in their urine, especially related to prostatic cancer, etc. But you can have like normal red colored urine as well, and it's just part of whatever you're eating in your diet or different types of drugs. Okay, so what I'm going to do is I will record the rest of today's lecture and Wednesday's lecture, so there won't be any formal class on Wednesday. You can stay home and listen to the recording. It'll be available for you. Hopefully it'll be of sufficient quality, and I'll use the little laser pointer. And then we'll have class again on Monday. So make sure that if you have any questions to let me know. The one thing I'm going to ask you to think about with your colleagues, because I need to put these questions together by this weekend, and you can let me know on a discussion page that we'll set up on your course website which of those 4 different sections you would want to have tested, and 3 of them be okay. Have a great week, and I'll see you all next week on 11 p.m.